warahmatullahi wabarakatuh, and hi everyone. I hope all of you will feel always good and stay safe at home. In these episodes, I would like to share with you the students' further investigation, which become the project-based learning in Chapter 4, Metabolism and Enzyme. The aim is to investigate the action of catalase on hydrogen peroxide. At the end of the investigation, students should be able to identify what is hydrogen peroxide and why it is produced in the body cells, why it is dangerous to our body cells, what enzyme we have in our body cells to catalyze the breakdown of hydrogen peroxide, explain the process of breakdown of hydrogen peroxide, and prove it. Compare raw and cooked liver and potato cells reactions and explain. Justify moral values and the advantages of God creates the speciality of living organisms. And finally, make a conclusions. Remember, biology always study for life. So let's find out.
Hi everyone, I am Alia from 4 Step 2. Today I'm going to do an experiment for biology subjects. The topic that we are learning now is metabolism and enzymes. In the liver catalase, there is an enzyme called catalase. So today I am going to do an experiment about catalase enzyme. The aim for this experiment is we are going to see what's the reaction of hydrogen peroxide or H2O2 to the chicken liver and potato. What is H2O2? Hydrogen peroxide is a simple compound. Its physical properties are that it's a liquid but more viscous than water. The formation of hydrogen peroxide in our body is to protect the body from certain harmful substances. For example, when the immune system is activated to respond to the bacteria, large amount of hydrogen peroxide is produced by certain cells to fight with the infection. Yes, drinking high concentration of hydrogen peroxide can be very dangerous because it can cause tissue to burn. Another example is when hydrogen peroxide is swallowed, it can generate bubbles in our stomach. There can be a lot of stomach irritation and even burns that require a trip to the emergency room. Luckily, there is an enzyme called catalase that helps to protect our body from oxidative damage cell by breaking down the hydrogen peroxide to water and oxygen. I will show you how the process works. Put raw chicken liver in a container A. Then, pour 40 ml of hydrogen peroxide and close it. See, the wooden splinter is burning because this process produces oxygen. The wooden splinter is not burning because not enough oxygen. The wooden splinter is not burning because this process not produce oxygen. The wooden splinter is not burning because this process not produce oxygen. Hydrogen peroxide react to the raw chicken liver and raw potato by producing oxygen and bubbles. Raw chicken liver and raw potato have catalase enzyme so that they react to the hydrogen peroxide. But boiled chicken liver and boiled potato does not react to the hydrogen peroxide. The chemical reaction, which is the bubbling, will not take place because boiling would destroy the enzymes. Moral of the story: every creation of God was so beautiful. God created something definitely wholesome. For example, hydrogen peroxide is needed in our body, but at the same time, it is also dangerous. Therefore, there are enzymes that help to restore the condition. So we need to appreciate it and take a good care of it. In conclusion, biology is study for life. Without biology, we don't know about the health of our bodies, our pets, plants, and the environment in our home. We may not even know which one is medicine and which one is poison. So, study biology to save your life. Bye!
Today, I'll be carrying out an experiment to investigate the action of catalase. Before we start, I'd like to tell all of you about catalase and hydrogen peroxide. What is catalase? Catalase is an enzyme present in the cells of many plants and animals. Cells with a high metabolic rates have a high concentration of catalase. This is the following equation that involves catalase. As you all can see in the equation above, it catalyzes the breakdown of hydrogen peroxide to water and oxygen. Now, I'd like to tell all of you about hydrogen peroxide. Hydrogen peroxide, or better known as H2O2, is well known as a cell-damaging agent that is produced during normal cell metabolism of aerobic organisms. For example, when the immune system is activated in response to bacteria, large amounts of H2O2 are produced by certain cells to fight the infection. Why is H2O2 dangerous to our body cells? This is because it is an excessive production of oxygen metabolites, which leads to oxidative stress and diseases. Now, if the level of H2O2 becomes too high, the cell's DNA and other proteins can be damaged. Scientists suspect that this and similar processes are what leads to cancer, diabetes, and many more diseases. Now that I've explained about catalase and hydrogen peroxide, let's get started with our experiment. So here are some materials and apparatuses you need for this experiment. One of them is the hydrogen peroxide, but of course we won't be using all of them. We'll be only using a small amount. A piece of chicken liver, one raw, one boiled, a potato, one raw, one boiled, 4 containers labeled A, B, C, and D. Finally, a stick from a penyapu lidi, which you can use it as a glowing splinter if you use it with a match. Before that, remember to wear your gloves and of course the face mask. Let's get started. Alright, so first I'm going to take all the food over here and I'm going to cut them into half. Now the reason why am I doing this is because we don't want to waste the material, so that's why I'm gonna cut all of the food into half. I'm going to place the food into their respective containers. First, we'll start with the raw chicken liver. The other half we're just gonna put it back here. Alright, fast forwarding from here. Some hydrogen peroxide into the containers. And remember, be very, very careful. Oh! Okay, there is no reaction happening here. Also here. Now we're going to be using a glowing splinter to test the presence of oxygen in each of the containers. Alright, so we'll start with container A. Alright. As you can see, it burns vigorously. So there's actually a lot of presence of oxygen in there. Alright, now container B. Oh, it burns a little bit. We're gonna put it in container C and we're gonna see what happens. Oh, it burned off completely. So this means that there's actually no presence of oxygen in here. And now we're gonna put it in D and we'll see what's gonna happen. And yes, it burned off completely again. And just like say, there is actually no presence of oxygen in here. After doing the experiment just now we all know that hydrogen peroxide reacts to catalase, which are present in both the raw chicken liver and the potato, causing an oxygen from H2O2 to split off, creating the bubbles we see. As you'll notice, the cooked chicken liver and potato produced from little to no bubbles. And this is because the heat degraded the catalase, making it incapable of processing the hydrogen peroxide. So that's all from me today. Thank you so much for watching this video. I hope all of you have learned a thing or two from this experiment, and always remember, biology is study for life.